Good morning, good afternoon, or good evening, depending on where you are in the world. I'm delighted to welcome you to the latest Novagene webinar, Next Generation Sequencing Solutions for Transcriptomics and Epigenetics Research. I'd like to thank you all for taking the time to attend today's webinar. As ever, we've got a lot of you here and we're looking forward to receiving your questions. A bit of housekeeping before we start. The webinar will last for around 45 minutes with 15 minutes for question and answers at the end. Please submit your questions via the Q&A button at the bottom of your screen. You can upvote a question by clicking the thumbs up symbol next to it. We'll try and prioritize the most popular questions. If we don't have time to answer your question, we will come back to you via email after the webinar. Please don't use the raise hand functionality. Just send any questions via the Q&A or message us directly if you have a technical issue. We'll share the slide deck and a recording of the webinar via email. You can expect this on Friday. There'll be a link to view and print a certificate of attendance for any students on the webinar who require one. Note you'll need to save or print this directly from your browser. Unfortunately, we can't provide this as a document. We'd love to hear your feedback, particularly around how we can make these webinars more useful to you. We'd appreciate it if you could take a couple of minutes to complete the survey that will pop up after you leave the webinar. I'd now like to introduce your host for the webinar as Selvi Ergin, who is our UK account manager for the East of England. Selvi, over to you.
Thank you very much, Joseph. Nice introduction for me. Yes, I would like to introduce myself too. My name is Selvi and I am an account manager based on East of England. So today I'm going to go through a brief overview of Novagene. Novagene was founded in 2011 and has been growing rapidly since then. Our company Novagene mission is to improve life science, clinical research fields, diagnostic, and human healthcare. At Novagene, we have expertise in a variety of different areas, such as agriculture, biopharma, genetic health. As you see here, through this picture, our work covering the main aspect of NGS wide variety of application. Our European Sequencing Center is based on the Cambridge Science Park in UK. Here we have state-of-the-art lab equipment with the latest NovaSeq 6000 platforms, which allow us to provide a fast turnaround time and high quality data. We also have supercomputer cluster here. We can offer bioinformatics services and to store the data in the UK. Not only do we have a lot of experience, but also we are a global presence around the world. So we are a global company. We can offer services across Asia and the US and of course Europe. We have labs in USA, UK, Singapore and China. We are almost 2,000 employees and serve over the 5,600 customers in the world-class universities, biotech companies and hospitals. We can provide sequencing from small to large projects. We provide a full range of sequencing services that covers a wide range of applications from human genome, transcriptome, microbial genome, epigenome, or animal for plants. So, today, Dr. Hamid's presentation will deal with transcriptomics and epigenetics research. In this free webinar, we will learn about the current methodologies in the transcriptomics and epigenetics, sample types, and recruitment for various applications and NGS solutions and analysis approach. NGS market is really crowded, so you can ask why Novogene. There are a lot of good reasons why you should trust Novogene with your projects. When we ask our customers for the top reasons why they choose to work with us, they mention data quality. I would like to remind that we pride ourselves on doing the best quality data for you and ensuring excellent quality. The way we do that is through quality control checks. They also mention capacity that we have and, of course, faster turnaround time. Furthermore, the analysis that we can provide you. In terms of the technology, we only use the gold standard products, and that is why we have partnership with Illumina, and we use their latest platforms, which is Nova 6000. We are proud of our record in the scientific community. We have been published in over the 790 research papers with a total impact factor over 4,000 is about 5,000. We are also driving the NGS industry forward with over 37 NGS-related patents. So, our technology is helping to make NGS process more efficient and effective, all the while improving the quality of the data we deliver. Not only do we pride ourselves on quality and some things that Novogene is very good at, we also invest a lot in people like myself, account manager as well. We want to ensure that we provide you with the best help and guidance to meet your research goals in the best way possible. We have local account managers in the different countries. So the Novogene community keeps on growing. Our expert team are located across Europe and are ready to assist you in your next projects. Here we are in all of our glory. I'm going to keep these slides open to know your local account manager from UK, Ireland, stack. With that, I would like to thank you for your attention, and I will hand over to Dr. Reid Hamidi. He is a product manager at Novogene. He is responsible for carrying out the market analysis and upgrading current products and the services, the field of next generation sequencing at Novogene. Moreover, Dr. Hamidi delivers technical training and develops various market promotion activity. Thank you very much.
Thank you, Selvi, for the nice introduction. I'll share my screen now. Okay, I think everything should be fine. Good. So hello, everyone, and welcome again to today's webinar, where I'm going to talk about NGS solutions for transcriptomics and epigenetics research. Before I start, this is the outline of the webinar for today. In the first part, I'm going to talk about epigenomics, including DNA methylation, histone modifications and transcription factors, and chromatin accessibility. After that, I'm going to move on to transcriptomes, including messenger RNA, long non-coding RNA, and small RNAs. Towards the end of the webinar, I'm going to show you different case studies where several epigenomic and transcriptomic solutions were combined together. So to start with, epigenomics by definition is the study of heritable changes in gene function that do not involve alterations in the DNA sequence itself. So unlike genetics, where changes in gene expression come from mutations that would affect the DNA sequence itself, epigenetics includes all the changes within gene expression that come from alterations that do not change the DNA sequence itself. Research focusing on epigenetics in the last couple of decades have been able to identify three main characteristics of the system. First of all, the system must be mitotically and or meiotically heritable. In other words, epigenetic information can be transmitted from the mother cell to the daughter cells during the process of cell division and mitosis. In addition to that, epigenetic information can also be transmitted intergenerationally from the parents to the offspring. In addition to that, the system must be self-perpetuating on its own, and more importantly, it is reversible. In other words, any epigenetic changes that happen can be changed or reversed at a later point in time. Among the most prominent epigenetic events that take place within the cell, we have DNA methylation affecting the nucleotides that make up the DNA molecule, and we have histone modifications, transcription factors, and chromatin accessibility affecting the proteins that are associated with the DNA. Starting with DNA methylation, out of the 4 nucleotides that make up the DNA molecule, only adenines and cytosines can be methylated. Cytosine methylation is widespread in eukaryotic and prokaryotic organisms. However, it varies greatly between species. It can be up to around 14% in Arabidopsis thaliana or as low as 0.03% in Drosophila class. On the other hand, adenine methylation has been observed in bacterial, plant, and mammalian genomes, but is less investigated than cytosine methylation. In plants, DNA methylation can be found in different contexts. It can occur at the level of CpG dinucleotides, or CHG, or CHH, where H corresponds to either A, T, or C. However, in mammals, DNA methylation is almost exclusively found at CpG dinucleotides at both strands of the DNA molecule. Interestingly, several papers and reports have hinted that non-CpG methylation can also occur in mammals, but at very specific contexts, including embryonic stem cells or during neuronal development. In terms of function, DNA methylation has been implicated in a lot of cellular processes and developmental stages in humans. For example, DNA methylation is involved in aging, in embryonic development, in exercise, and cancer. More recently, DNA methylation has been implicated in immunity, psychology, heart disease, and even in metabolism. Similarly, DNA methylation plays important roles in plants, especially during their growth and in their environmental resistance to different stress signals. In animals, DNA methylation has also been shown to be important for their development and their behavior. Now, talking in details about DNA methylation, and in particular 5-methylcytosine, we know that in mammals, 5-methylcytosine is the most common form of DNA methylation. Several reports have shown that it can affect up to 70 or 80% of the CpG islands in humans. And in terms of functional consequences, we know that CpG island methylation in the promoter region of any specific gene is usually associated with repression and loss of gene expression. More importantly, 5-methylcytosine is maintained during the process of DNA replication. In other words, the information of 5-methylcytosine is faithfully transmitted from the mother cell to the daughter cells during the process of cell division and mitosis. Researchers that are interested in studying 5-methylcytosine rely on a method called whole genome bisulfite sequencing. In fact, whole genome bisulfite sequencing is considered the gold standard for investigating DNA methylation. And one of the main advantages of this method is that it gives us the ability to measure methylation at the level of single cytosines. During the process of bisulfite conversion, unmethylated cytosines are exposed, and through the bisulfite treatment will be converted to uracil. On the other hand, methylated cytosines are protected from this conversion and will remain as cytosines. During the whole genome bisulfite library preparation, the traditional method has been as follows. First of all, we start with the genomic DNA from the cells that we are studying. After that, the genomic DNA is fragmented into smaller pieces to make it compatible with next generation sequencing. Afterwards, sequencing adapters are added, followed by bisulfite conversion in order to convert unmethylated cytosines to uracil. In the end, the DNA fragments are purified, and then limited PCR amplification is performed in order to generate the sequencing library. However, in the last couple of years, several other methods that are different from the traditional library preparation method have been developed. One of these methods is called post-bisulfite adapter tagging, or PPAT. In the PPAT workflow, what we start with is, again, the genomic DNA, followed by fragmentation and bisulfite conversion. And in the end, we have the sequencing adapter tagging and later PCR amplification. PPAT has a lot of advantages over the traditional library preparation method because, first of all, it requires less genomic DNA as input material. And more importantly, the bisulfite conversion step does not affect the quality of the library, since bisulfite conversion is carried out before the sequencing adapters are added, unlike the traditional method where the adapters are added first and then bisulfite conversion is done. Now, after preparation of the sequencing library and next generation sequencing, what we will have is the sequencing reads. When we get the sequencing reads and the raw data, the first step that we do is to perform data QC in order to make sure that the sequencing reads are of good quality. Afterwards, we perform Bismarck alignment, where we align the bisulfite treated reads to the reference genome of the species that we are studying, such as human or mouse. After Bismarck alignment, we perform 5-methyl cytosine calling in order to identify the cytosines that have been methylated in our sample. Once we know the information about the methylated cytosines in the genome, we can perform different types of analysis. We can look at the methylation level at different levels. In these two graphs here, what we can see is the methylation density in a global chromosomal level, where we can see the DNA methylation profiles across the whole chromosome within different chromosomes. Similar type of analysis can also be performed at the level of gene elements, such as promoters, exons, introns, or UTR regions. If we are comparing different samples to each other or different conditions, we can perform differential methylation analysis. This type of analysis can be performed at the level of the chromosomes in order to see which regions of the chromosomes now have gained DNA methylation or lost DNA methylation between the different samples or the different conditions. Similarly, the differential methylation analysis can also be performed at the level of functional regions, including exons, introns, promoters, repeats, or UTR regions. So now that we have covered DNA methylation and 5-methyl cytosine in particular, in the next part, I'm going to talk about histone modifications, transcription factors, and chromatin accessibility. Researchers that are interested in histone modifications and transcription factors rely on a method called ChIP-seq. ChIP-seq stands for chromatin immunoprecipitation with DNA sequencing, and as a method, it has been developed in order to analyze protein interactions with the DNA. The main aim of the method is to identify the binding sites of DNA-associated proteins. ChIP-seq can be used with both histone modifications and transcription factors, including the RNA polymerase machinery. One interesting thing about histone modifications is that they are post-translational modifications, such as methylation, acetylation, ubiquitination, or phosphorylation, that can take place at the different amino acids constituting the histone tails of the histones that are incorporated within the nucleosome, such as H2B, H2A, H3, and H4. The different consequences of these histone modifications depend on which type of histone modification is being deposited, and the final outcome will be either an increase in gene expression or a loss of gene expression. In terms of the ChIP-seq workflow, as I have mentioned earlier, what we are interested in here is DNA binding proteins and determining the specific binding sites of these proteins throughout the genomic DNA. In a ChIP-seq experiment, the first step is to perform cross-linking with reagents such as formaldehyde. The importance of cross-linking here is to lock in or to stabilize these interactions between the proteins and the genomic DNA. Afterwards, the genomic DNA is fragmented into smaller pieces that would make it compatible with next generation sequencing. Afterwards, an immunoprecipitation step with a protein-specific antibody is carried out. During this step, we rely on an antibody that is able to recognize the transcription factor or the histone modification that we are studying. And during that immunoprecipitation step, the DNA that is bound to the protein or the histone modification will be pulled down, while other DNA fragments that are not bound by the protein or the histone modification will not be immunoprecipitated. Once this immunoprecipitation is done, and these immunoprecipitated DNA fragments along with it, we perform DNA purification, and then we add the sequencing adapters to generate the sequencing library. Following next generation sequencing and data collection, we take the sequencing reads and we align them to the reference genome of the species that we are studying, such as mouse, human, or Drosophila, in order to identify the peaks throughout the genome that correspond to the binding sites of the transcription factor or the histone modification that we are studying. So for the ChIP-seq analysis pipeline, as I have just mentioned, the first step to do is to align the reads to the reference genome of the species that we are studying. After the alignment, we perform peak calling in order to identify the regions of the genome that are bound by our transcription factor or the histone modification that we are studying. We can perform this at the level of different samples. And in reference, we have an input sample that we use in order to normalize our sequencing needs. Once we have identified the regions that are bound by the protein, we can perform different types of analysis. The first one is motif analysis, where we are able to determine whether our transcription factor or histone modification has a bias or a tendency to bind to a very specific sequence throughout the genome. In addition to that, and after identifying the genes that are bound by our proteins, we can take these genes and perform different types of analysis in order to determine whether they are involved in a biological process or a cellular pathway within the cell. Moving on to the next method that we use here, which is ATAC-seq. ATAC-seq stands for Assay for Transposase Accessible Chromatin Using Sequencing, and as a method, it has been developed in the last couple of years to assess genome-wide chromatin accessibility. In fact, ATAC-seq has a lot of advantages over other methods such as mRNA-seq or DNA-seq because it is faster and way more sensitive. The principle of the ATAC-seq experiment relies on a hyperactive TN5 transposase that is able to insert sequencing adapters into the open chromatin regions throughout the genome. On the other hand, closed chromatin regions will not be targeted by the TN5 transposase. This is an overview of the ATAC-seq workflow. So in the first step, the enzyme is added to the sample where it will target the open chromatin regions throughout the genome. Afterwards, the enzyme will start cutting these DNA fragments of the open chromatin regions, and at the same time, it will add sequencing adapters so that these DNA fragments are compatible with next generation sequencing. Afterwards, the DNA fragments are purified, and with limited PCR amplification, we obtain our sequencing library. By performing next generation sequencing and data analysis, what we will have in the end is sequencing peaks that correspond to the open chromatin regions throughout the genome, while no peaks will be scored for the closed chromatin regions. So by now, we have covered epigenomics, focusing on DNA methylation, affecting the nucleotides of the DNA molecule, but also histone modifications, transcription factors, and chromatin accessibility, affecting the proteins that are associated with the DNA molecule. For the next part, I'm going to focus on transcriptomics, and we'll talk more in details about different RNA molecules and how they are involved in regulating gene expression. In general, RNA molecules within the cell are divided into two main categories. The first one is the messenger RNA that is responsible for encoding for proteins that are essential for the survival and the functioning of the cell. Everything else is considered as non-coding RNA because these RNA molecules are not able to encode for proteins. Non-coding RNAs are further subdivided into different categories depending on their function and their length. On one hand, we have tRNAs that are responsible for linking the mRNA to amino acids during the process of translation. We also have ribosomal RNAs that are responsible for forming the ribosome. Interestingly, ribosomal RNAs are the most abundant RNA molecules within the cell, where they can account up to 80 or 90% of the total RNA within the cell. On the other hand, we have long non-coding RNAs that are responsible for regulating gene expression, and small RNAs, such as microRNAs, also involved in regulating gene expression, smaller nuclear RNAs involved in RNA splicing, and siRNAs involved in silencing gene expression. The first RNA that I'm going to talk about is the messenger RNA that is responsible for encoding proteins. During the process of transcription, the DNA sequence is transcribed into a single-stranded RNA molecule that has the information coming from the exons, the introns, and also with a poly-A tail at the 3' end. During the process of alternative splicing, the intronic regions are removed and different combinations of the exons can be formed in order to give rise to different proteins or different isoforms of the same protein during the process of translation. Researchers that are interested in studying the mRNA within a particular sample or a tissue rely on a library preparation method that includes poly-A enrichment. So as I mentioned earlier, almost all of the mRNA within the cell has a poly-A tail. And this is the method that we rely on to enrich for the messenger RNA. After that step, we perform fragmentation, followed by reverse transcription and the second strand cDNA synthesis. Once we have the cDNA, we add the sequencing adapters, and with limited PCR amplification, we have a sequencing library that is compatible with next-generation sequencing. After performing next-generation sequencing and collecting the data from the RNA-seq experiment, there are two main pieces of information that we can obtain here. The first one would allow us to identify the messenger RNA that is present in our samples. So we are able to determine which mRNA species are present within a particular sample. In addition to that, we are able to perform a quantification and identify how many or how abundant each mRNA is within the cell. So for the analysis pipeline, as I have just showed you, the first step is to perform a quantification where we are able to determine the expression level of each gene within a particular sample. If we have different samples or different conditions and we compare them to each other, we can perform expression pattern analysis. And this will help us to identify genes that are being either upregulated or downregulated. In other words, we are able to shortlist the genes that are changing their expression between the different samples or the different conditions by either increasing their gene expression or by decreasing the gene expression levels. Once we have shortlisted these differentially expressed genes, we can perform different types of functional analysis where we can investigate whether these genes are involved in a particular cellular process or a biological pathway within the cell. Now, moving away from the messenger RNA and into the non-coding RNAs, the first one I'm going to talk about is the long non-coding RNA. By definition, long non-coding RNAs are non-coding RNAs that are longer than 200 in nucleotides, and they are not able to be translated to proteins. Depending on the genomic position from which the long non-coding RNA is transcribed, they can have different names. They can be either intergenic long non-coding RNAs, intronic, sense, antisense, or even bidirectional long non-coding RNAs. For researchers that are interested in investigating the long non-coding RNAs within a particular sample, the library preparation method that we rely on here is a directional library preparation. So following enrichment and fragmentation, we perform reverse transcription followed by second strand cDNA synthesis. The main difference here in the long non-coding RNA pipeline to the messenger RNA pipeline that I showed you before is that during second strand cDNA synthesis, instead of using dTTP, we use dUTP. After obtaining the cDNA, we add the sequencing adapters to the cDNA fragments. However, before performing PCR amplification, we digest the DUTP second strand, and this will help us obtain the directionality of the long non-coding RNA. In other words, we are able to determine later during data analysis whether the long non-coding RNA was transcribed from the sense or the anti-sense strand of the DNA molecule. In terms of data analysis, the first step that we perform after sequencing the reads is a filtering step in order to enrich for the long non-coding RNAs. This is done using different programs. After the filtration step, we can actually look at the distribution of the reads throughout the genome so that we can also obtain information from which regions of the genome these long non-coding RNAs are transcribed from. Another filtering step that we can perform here is the coding potential filtering in order to determine the coding potential of the different long non-coding RNAs that we have performed. Furthermore, what we can also perform here is actually novel prediction of new long non-coding RNAs that have not been reported before in the literature. So we are able to determine new long non-coding RNA species that are present in our sample or that have not been reported in any study before. Now, research focusing on long non-coding RNA have been able to identify one of their major functions as cis-regulatory factors by recruiting remodeling factors to local chromatin. So what is happening here is that long non-coding RNAs are able to interact with the transcription factors and mRNA in order to regulate gene expression. And because of this, it is very important to study the co-expression between the long non-coding RNA and the messenger RNA. We perform this type of co-expression analysis between the long non-coding RNA and the mRNA according to a set of thresholds and parameters that we specify. In this case, the parameters that we rely on are a Pearson correlation that is higher than 0.7 and a p-value that is lower than 0.05. Using this co-expression information, we are able to build an interaction network of all the different long non-coding RNAs and the messenger RNAs within the sample. In this summary here, in this table summary here, I'm showing you how different long non-coding RNAs are being co-expressed with their respective messenger RNAs according to the thresholds that we have set, including a Pearson correlation that is higher than 0.7 and a p-value that is lower than 0.05. Now, moving on to the other non-coding RNA species, the first one I'm going to talk about here is microRNA. By definition, microRNAs are short, single-stranded non-coding RNAs that are around 22 nucleotides in length. It has been shown that the human genome encodes for around 2,300 microRNAs, and in terms of function, they have been shown to be involved in RNA splicing and post-transcriptional regulation of gene expression. On the other hand, circular RNAs are circular single-stranded non-coding RNAs where the 3' and the 5' ends are joined together in order to form a continuous loop. Interestingly, many circular RNAs can arise from protein coding genes, but their biological function remains unclear. However, recently, some papers and some studies have hinted that circular RNAs are involved in gene regulation and cancer. Now, for the microRNA analysis pipeline, the first step that we do here after collecting the sequencing data is to perform length filtering, depending on the length of the RNAs. So as I told you earlier, microRNAs on average are 21 to 22 nucleotides, which are shorter than other RNA species in the sample, including siRNAs that are around 24 nucleotides and piRNAs that are between 28 to 30 nucleotides. So after performing length filtering to enrich for the microRNAs, we can now identify novel microRNAs that were not reported before in the literature. This is done in a stepwise process, starting with the non-microRNAs and then moving on to the ribosomal RNA, the tRNA, the repeats, until in the end, we have shortlisted novel microRNAs that are specific for the sample that we are studying and that were not reported before. Now, once we have the information from both the non and the novel microRNAs within our sample, we can then investigate the occurrence of different miRNA families within the sample in a species-specific manner. So what I'm showing you here is an example where different microRNA families have been identified for a human sample. Now, in terms of function, circular RNAs have been shown to act as microRNA sponges in order to inhibit the repression of gene expression. So they are able to interact together, and the circular RNAs in this case are acting as sponges in order to inhibit the repression of gene expression that is carried out by the microRNAs. And because of this, it is very important to analyze both microRNAs and circular RNAs within our samples. This can be performed with different softwares depending on the species that we are studying. We use Miranda for animals and PS Robot for plants. And what we will obtain from the analysis is a prediction of the microRNA binding sites for each circular RNA molecule within our samples. So through this, we are able to predict and identify the miRNA binding sites of the different circular RNAs within the sample. Now, something interesting that you might not be aware of or something that you have come across recently is that several reports have hinted for possible interactions between different RNA species within the cell. And these interactions have been implicated in diseases such as musculoskeletal degenerative diseases or pulmonary fibrosis or even diabetes. More importantly, similar types of interactions between the RNA species have been reported for the SARS-CoV-2 infection causing the latest pandemic. So in this case, we know that different RNA molecules are involved in a complex network of interactions and competitions within the cell. So what if we are actually interested in having a global map or a global overview of all the different RNA molecules present within the sample and to investigate the different interactions and the competitions that are happening? Our answer for this is to perform whole transcriptome analysis. What we perform or what we can offer in this case is that we take the total RNA from the sample and then we split that into two main parts. The first part will undergo ribosomal RNA removal and a directional RNA library preparation. The information that we obtain here will help us in identifying and investigating long encoding RNAs, messenger RNAs, and circular RNAs. On the other hand, the other half of the total RNA sample will be used for small RNA library preparation, and this will give us information about small RNAs. After sequencing and combining the data from all of these different RNA molecules, we can perform the joint analysis that will help us in having a global overview of the different RNA molecules within the species, within the sample, and the different interactions and competitions that they might have. In this table here, I'm summarizing the library types for the different RNA species. So starting with the messenger RNA, the standard library preparation relies on ribosomal RNA removal, followed by poly-A enrichment. The library can be either directional or non-directional. In case our starting material is RNA from blood samples, then in this case, we have to rely on globin and ribosomal RNA removal. and the library is directional. For the long non-coding RNAs, the library preparation relies on ribosomal RNA removal and is directional. For the circular RNA sequencing, the library preparation relies on ribosomal RNA removal, it is directional, and most importantly, there is a linear RNA removal step because we are interested only in the circular RNAs, so we need to get rid of any linear RNA within the sample. And finally, for the small RNA sequencing, we will enrich for RNAs that are on average between 18 to 40 base pairs in length. So now that we have talked in details about epigenomics and transcriptomics, including the different RNA species, in the next part, I'm going to show you different case studies where various epigenomic and transcriptomic solutions were combined together in order to investigate the regulation of gene expression. The first paper that I'm showing here from 2020, the researchers were interested in studying the effects of histone modifications on gene expression during mouse spermatogenesis. So in this report, the authors performed ChIP-seq experiments for different histone modifications to see the overall binding profile of these histone modifications throughout the genome. And then in the second part, they performed RNA sequencing experiments in order to determine the expression levels of the different genes throughout the genome. By combining the data from the ChIP-seq experiments and the RNA-seq experiments, the authors were able to identify different clusters of genes that are involved in different cellular processes and pathways during mouse spermatogenesis. In the second example here, also a paper from 2020, the authors investigated DNA methylation levels and oncogene expression levels during cancer pathogenesis. So in this study, the authors performed whole genome bisulfite sequencing to determine the DNA methylation levels in both normal and cancer tissues. In parallel, they looked at the gene expression profiles by performing RNA sequencing experiments, again for normal and cancer tissues. By combining the DNA methylation data with the gene expression data, the authors were able to identify that genes that are hypomethylated in the promoter region, in other words, genes that have very low DNA methylation levels in the promoter region, actually have high expression profiles. And the opposite is true for the other genes that are now hypermethylated in the promoter region, which means that they have high DNA methylation profiles, and looking at the expression data, these genes, as expected, have low gene expression. For the last case study that I'm going to show today, the authors used different methods in order to dissect the interplay between DNA methylation and gene expression that is mediated by RNA polymerase 5 in Arabidopsis. So in this study here, the authors relied on DNA methylation experiments to investigate DNA methylation within different contexts, such as the CG dinucleotides, CHG, or CHH. In addition to the DNA methylation, they performed the ChIP-seq experiments for RNA polymerase 5 in order to identify the regions of the genome that are bound by the transcription machinery. In addition to that, they performed RNA sequencing experiments in order to investigate the gene expression levels of the genes that are located downstream of the DNA methylation and the polymerase 5 binding sites. And this really highlights the amount of information that we would get by combining different transcriptomic and epigenomic solutions together. So with this, I come to the summary slide of today's webinar. So what I have showed you today is that regulation of gene expression is a very complex process and involves both epigenomic and transcriptomic events. For the epigenomic events, we talked about DNA methylation that we can investigate using whole genome bisulfite sequencing. We talked about the distribution of histone modifications or the binding of different transcription factors to the genome, and this can be assessed by performing ChIP-seq experiments. And finally, for the epigenomics part, we talked about the chromatin accessibility that can be measured using ATAC-seq. In parallel, for the transcriptomics, we discussed different RNA-seq experiments that would allow us to identify and evaluate the expression levels of different coding and non-coding transcripts. All of these epigenomic and transcriptomic events and interactions within the cell are very important to regulate gene expression within a particular sample. And with this, I would like to thank you all for joining us today, and I'm super happy to answer your questions.
Thank you very much for a nice presentation, Dr. Hamid. So we have had a lot of questions, so we are going to try and go through this in the meantime, and we have been allocated. So I'm going to move on to the questions and answer now. I'm going to start off with a question from Peter. And it says, will whole genome be sulfide sequencing detect methylation and the CG, CHH, and CHG in mouse samples?
Yes, so whole genome bisulfide sequencing is able to determine the methylation, the cytosine methylation levels in different contexts, including CG dinucleotides, CHH, and CHG. But keep in mind that mouse is a mammalian species, which means that most of the methylation in this case will take place at the CG dinucleotides and to a lower extent at the CHH and CHG. But still, we are able to determine this information from the whole genome bisulfide sequence.
Okay. And we have another question from Kinga. And it says, hello, can I perform chromatin immunopreservation experiment without fixation in the beginning?
Yes, this is possible. In this case, we refer to the ChIP-seq experiment as a native ChIP-seq. So we don't rely on any reagent to fix the samples. But I would like to stress here that in this case, we are not stabilizing the interactions between the proteins and the DNA. And eventually, any weak interactions that are happening in the sample, the information might be lost during the ChIP-seq experiment. So the recommendation here is always to fix the samples in order to stabilize these weak interactions between the proteins and the DNA.
Okay, another one from Rebecca, and it says, can ChIP experiment be performed on specific histone modification in Drosophila cells?
Yes. So histone modifications in general are evolutionary-wise very conserved between different species. And by using different antibodies that are able to recognize different histone modifications, we can use Drosophila cells or Drosophila tissues to perform the ChIP-seq experiment. So yes.
Okay, this one's from Sarah. And she asks, is an input sample needed for ATAC-seq?
So for an ATAC-seq experiment, unlike the ChIP-seq, we don't need an input sample, because the way that the method has been developed is that we will have sequencing reads that will come only from the open chromatin, while the closed chromatin will not have any sequencing reads in the sample. And this can be used in order to normalize the sequencing reads. So the input sample is already there within the sample that we are, within the experiment that we have performed. So no, an input is not needed in this case.
We have another question from Maria, and she asked, how many replicates do I need for a ChIP-seq experiment?
Yeah, so for ChIP-seq experiments, the minimum that we can use is 2 biological replicates in order to make sure that the interactions and the peaks that we have scored throughout the genome are reliable and they are not coming from non-specific binding of the protein or the antibody that we have used during the ChIP experiment. The more, the better, but ideally at least 2 biological replicates should be enough to remove any any bias in the data or any artifacts.
And we have another from Anjana. Hi, could you please specify why does peaks mean for cheap and ataxic? Does it mean the same in both?
Okay, yeah, that's a good question. Many people can get confused about that. So I will just go quickly back to the slides to show the difference, because we call them peaks in both cases, but they are not the same. So when we talk about the ChIP-seq experiment, the peaks that we score here, they correspond to the DNA binding site of the protein or the histone modification. In other words, they are these specific sites. So if we look at this genomic DNA, this binding site here will have a peak so that we know, okay, the protein or the histone modification is present at this particular region, while other regions that are not bound will actually be flat without any sequencing reads coming from that part. So in a ChIP-seq experiment, a peak refers to the binding site of the protein or the histone modification. However, in an ATAC-seq experiment, the peak here does not refer to a binding site, it refers to the open chromatin region that was targeted by the enzyme during the ATAC-seq experiment. I hope this makes it clear.
Okay, thanks. And the question is, does PBAT analyze whole genome?
Yes. So what we have in a PBAT experiment, I can go back to that slide. Exactly. So in this case, we start with the genomic DNA, and then we will have the total genomic DNA as an input material in this case. And then at the end of the day, any cytosine throughout the genome that is not methylated will be converted into uracil, while the methylated cytosines will remain as cytosines. So the bisulfite conversion will actually convert any cytosine throughout the genome that is not methylated.
Okay. And this was from Anna, and she asked any specific introductions for stranded with unstranded sequences.
Okay, yeah. So for the RNA preparation, for the RNA-seq experiments, as I was showing in the slides, there are different methods that we use here. So for example, for the messenger RNA, because it is non-directional, what we do here during the second strand cDNA synthesis, we use regular nucleotide dNTPs, including dTTP. However, when we talk about a stranded or a directional library preparation, and this is the case of long non-coding RNAs, which we have here, what will happen, the difference between this and the previous one is that during second strand cDNA synthesis, instead of using dTTP, we use dUTP. And later, the second strand in the cDNA molecule that has the dUTP will be digested. And this, in a way, will preserve the directionality of the long non-coding RNA, or in other words, it will give us the information whether the long non-coding RNA was coming from the sense or the anti-sense strand of the DNA molecule. So it is a small modification in the protocol, but it really helps us in this case to determine the directionality of the long non-coding RNA.
And another question from Jelena, what method for DNA methylation sequencing do you suggest, PBAT or PRBS?
Yeah, so both of them can be used. It really depends on the biological question or the research aim that we have here. So in case if you are interested in studying the whole genome, then I would recommend PBAT, because it can really look at the whole genome. In case she is interested in specific regions of the genome, then I would recommend the reduced bisulfite sequencing in this case. So in conclusion, it really depends on the research objective. For a global genome-wide overview, we should go with whole genome bisulfite sequencing. For a more targeted or specific region methylation analysis, I would recommend the reduced bisulfite sequencing.
Okay. Great. Thank you. Like I said, we have so many questions, and unfortunately, we can't answer all of them, but we will get back to you. It might take you a couple of days. So that is for today. With this webinar, we are very happy to announce that we are having discounts to get up to 30% of library preparation and sequencing for the eukaryotic mRNA-seq, eukaryotic long-non-coding RNA-seq, whole genome bisulfite sequencing, and chip-seq. In the meantime, please stay in touch. You can follow us on Twitter and LinkedIn, or you can get in touch via our website at novagene.com. We have many service flyers in our hub, and also you can connect with us for sample recruitments with using this hub. Thank you again for your time today. We hope you enjoyed the webinar and found it useful. We will email you a link to the webinar recording and the slide deck, along with a short survey that we appreciate you taking the time to complete. This survey will help ensure we make future webinars even more relevant to you and your research goals. There will be a link in the follow-up email for you to view a certificate of attendance if you need one. We are looking forward to seeing you all the next webinar. Thank you again.